Hello, my name is Dr. Rahul Krishnan, and I'm a Gynecologic Oncology Fellow at the University of Chicago. In addition to my clinical work, I conduct research at the Ovarian Cancer Research Lab at the University of Chicago. My research interest lies in understanding how ovarian cancer develops. Each year, nearly 20,000 women are diagnosed with ovarian cancer in the United States. And despite promising new advances in recent years, it still remains the most lethal gynecologic cancer. When caught early, while the ovarian cancer is confined to the ovary, nearly 90% of patients survive their disease. Unfortunately, however, the vast majority of ovarian cancer cases are diagnosed at an advanced stage when the disease has spread to other organs. Even with chemotherapy and surgery, 5-year survival rates are less than 20%. No good screening tests exist for ovarian cancer, making early detection especially challenging. This is why learning more about how ovarian cancer develops is so important. In recent years, there's been increasing evidence that ovarian cancer may actually originate in the fallopian tubes. Yet very little is known about what causes normal fallopian tube cells to transform into pre-cancerous cells, and ultimately become ovarian cancer. The key to understanding the origin of ovarian cancer will be to understand what biological processes drive this transformation of normal fallopian tube cells to precancer and ultimately become ovarian cancer. My research is focused on understanding this process better. Thanks to generous support from the Krouse Rosenthal Foundation, we can conduct novel experiments to better understand the biology of what drives normal fallopian tube cells to transform into precancer and ultimately invasive cancer cells. To answer this question, we're utilizing two novel technologies that can study and characterize the RNA and protein activity in each cell type. The first is spatial proteomics, developed with our collaborators at the Max Planck Institute in Munich, Germany. The second is spatial transcriptomics, which lets us look at each individual cell type's gene expression. These technologies allow us for the first time to specifically measure and compare proteins and gene expression among the different types of cells in the ovarian cancer development process. Doing so will let us characterize and compare the molecular pathways that play an important role in the development of ovarian cancer. To date, we have already characterized the activity of 9,000 genes. The next phase of our study is to then test the effect of these genes on cancer biology by altering them in ovarian cancer cell lines in the lab. By doing so, we hope to 1. Understand how to prevent the development of ovarian cancer in the first place, 2. Develop markers that could help detect ovarian cancer early in its development, and 3. Potentially develop new treatments to better treat ovarian cancer.